Okay, let's get started. So welcome everyone to this monthly seminar of single cell and spatial analysis program. And it's my great honor and pleasure to introduce Dr. Sarah Teichmann. And Dr. Teichmann completed her PhD at the MRC Laboratory of Molecular Biology in Cambridge, UK, and started her research group there as well in 2001. During the years, she took on more and more leadership roles, including the head of cellular genetic program at the Wellcome Sanger Institute and the chair in stem cell medicine at the University of Cambridge. And she is the first faculty member appointed across both the EMBL, European Bioinformatics Institute, and the Wellcome Sanger Institute. And she was recognized by many prizes and awards. For example, the Fellow of the Academy of Medical Science and Royal Society. And her work was also recognized by many prizes as well. For example, the Lister Prize, Biochemical Society Walworth Medal, Amble Gold Medal, and so on. Her lab is at the interface between computational biology and the molecular biology, focusing on developing and applying cell atlas technologies to understand human tissue architecture. Dr. Teichmann co-founded the Human Cell Atlas Initiative, which aims to generate a comprehensive reference map of the types and properties of all human cells. And there's actually a Nature collection of many work under the Human Cell Atlas scope. And if you skim through that collection, you'll find out that Dr. Teichmann and her group contributed almost 1/3 of the work there. It's quite impressive. And she led the work for constructing cell atlas for like a variety, for actually so many different tissues, organisms, and systems, as well as computational work. And while I was reading those papers, I always wonder by myself, how could any researchers be so knowledgeable in diverse systems and produce so many impactful and fundamental work that it's going to benefit a lot of ongoing work for both biologists and computational biologists and many related areas. And with great enthusiasm, let's welcome Dr. Sarah Teichmann to talk about cells, tissues, and organs assembling the Human Cell Atlas.
Thank you so much. Can you hear me?
Yes.
Thank you so much for that kind introduction. It's a pleasure and an honor to be here today and to speak about mapping the human body. And I will share this screen. Can you see this in
Yes.
the right mode? Yeah, awesome.
Yes.
Great. Okie dokie. So what I'll talk about today is our journey through the Human Cell Atlas and then also applications to understanding disease. And, as you're all aware in this seminar series, I won't dwell on the resolution revolution in genomics, which is the ability to sequence the nucleic acid content of individual cells and nuclei. And working here in Cambridge, UK, I can't miss the opportunity to mention that the first single-cell genomics paper or single-cell transcriptomics paper that was published was by Azim Sourani at the Gordon Institute here in Cambridge, which is the Developmental Biology Institute. And what his group was interested in getting at were cells that were extremely rare in the developing embryo because their interest is in the development of primordial germ cells. And so what they did was handpick 4 or 5 cells from the early mouse embryo and then sequence the transcriptomes of those individual handpicked cells in a Nature Methods paper in 2009. And obviously their intent was to be able to study rare cells. But for many of us, what was very exciting was the prospect of being able to take a cell sample and then characterize it in a more high throughput mode and analyze the data in order to define the cell types and cell states in the sample in a data-driven way. And that's sort of what's indicated in this slide, taking a biopsy and then the data onto this two-dimensional sort of piece of paper, if you like, or this graph, and then defining the different cell types. And so that really became a reality in the years following that 2009 paper, with rapid development of technologies in the academic domain and also in the biotech sector, which basically made the generation of data sets more and more convenient and economical and high throughput. And hand in hand with that technology development, there's also development of spatial transcriptomics technologies, which had sort of sequencing-based kind of approaches with spatial transcriptomics and in situ sequencing, which you can which you can also based, and the imaging based basically with the Murfish in situ sequencing and related technologies. And so bringing together the development of the single cell genomics and the spatial, really it was clear that starting a consortium to map the cells in the human body was plausible, if you like. And so that's what, with Aviv Regev and about 100 other people, we met in London 2016 to launch an international project to create a comprehensive reference map of the types and properties of our cells. And the idea here is that we gain a better understanding of our tissue architecture, our organ function, anatomy, physiology, and so on, using this single cell resolution data at full molecular breadth. And of course, also develop a molecular medicine basis for diagnostics and drug development. And the way we designed the consortium is as a grassroots bottom-up initiative, and it's open to anyone to join at humancellist.org/join-HCA. And of course, there are members of your university who are contributing. I can see Sue Mahmood, on reproductive tissues, and basically a large community, that's now in over 100 countries with over 4,000 members that's contributing to this effort. And contributing can mean contributing biomedical expertise, samples, data, analytical techniques, et cetera, et cetera. So it's really a team effort, so to speak. And what we entered after 2016 was really a phase of establishing the community and generating data. And then where we're at now is at the same time integrating data sets and generating reference data objects, as well as moving towards further data generation to expand and complete the atlas with more samples, more tissues, more comprehensive coverage of the human body, and also more modalities, including, importantly, much more spatial data than we had in the first kind of iteration. And what I also want to emphasize is that we have, sorry, just get rid of that okay sign. That we're now, working on this computational integration exercise, and then I don't know, can you see this okay bar at the top, or should I?
The OK bar is not shown, at least in my screen. Probably that's more of like each individual laptop may have it or not have it.
Fine. Okay, that's good. Thanks. What we are releasing are reference data objects for each of the major organs and tissues that we have in our current bionetworks, of which there are 18. Each bionetwork has coordinators who are sort of leaders of that community, and then are important kind of arbiters in a sense of consensus cell annotation. So there's a computational component, but there's also a sort of scholarly knowledge, biomedical knowledge component in releasing these data objects. And so this is an ongoing effort with the lung and so on already released, and there are 14 more that are in progress. And there are over 100 million individual cells mapped from these 18 biological networks that are mostly healthy reference tissues, including also an embryonic and fetal development. And we also have an organoid bionetwork, which is in vitro models. So I'm going to stop the kind of introduction to the general project there and tell you a couple of stories from our own group, starting with one of the first organs kind of that's necessary to establish life, which is the placenta. And the question that really piqued our curiosity here is coming from an immune angle, because we were studying T cells and in mouse, but then wanted to understand in human, how is it that the maternal immune system can tolerate paternal antigens? Because of course, in the embryo and the placenta, which is embryonic derived, half of the antigens are maternal, but the other half are paternal, and so contain foreign antigens that should, under normal circumstances, be recognized as non-self. And of course, as you're probably aware, there are the trophoblast cells from the placenta basically enter into the uterus, which is called the decidua. So instead of the endometrium in the pregnant state, and then contribute to interactions with vessels for remodeling of arteries, to increase the blood supply for oxygen nutrients to cross the placenta and go into the embryo. And there are many, interesting kind of sort of very specific cell states and cell interactions that are associated with that tissue, with this highly kind of unusual situation of cells with foreign antigens mingling with the maternal cells. The way we tackled this question using the early cell atlas technologies that were available to us, SmartSeq and then later 10x, purifying cell populations, and then mapping them, kind of putting the pieces of the jigsaw puzzle together again with a sort of combination of prior knowledge from sort of classical pathology, with immunohistochemistry, with single molecule RNA FISH, and in overall that allowed us to map all the tissues, all the cell, the lineages, the different cell compartments, so the trophoblast, the immune cells, the epithelial cells, stromal cells, the vascular cells, et cetera, and contribute to understanding of this tolerogenic immune interface. And what we also wanted to understand is how the intercellular conversations are taking place at the molecular level. And to that end, what we did was collect basically the known cells, receptor cell surface, and in terms of adhesive or secreted ligands interacting with receptors in the plasma membrane, in order to computationally predict which cells were having sort of specific signaling conversations with each other. And one of the things that we were able to sort of shed light on was the different specific decidual NK cell states. So a natural killer cells would usually recognize if a cell doesn't have the canonical HLA, for instance, and through the KIR receptors on the surface and would kill. And these NK cells have very special cell states with and different levels of sort of activation or poising of their killing activity, with different levels of KIR receptors, both activating and suppressing, and of course interaction with special HLA, which was known before, but then also cell-cell interactions with these new sort of detailed cell states that we're able to show with the maternal decidual stromal cells and macrophages, and with enzymes that reduce inflammation by, for instance, breaking down ATP such as this ENT-PD1 enzyme on the cell surface of these decidual NK cells. So there are a whole variety of different mechanisms that are contributing to this cell state. They're actually in first trimester placenta. So we're studying most sort of the second half of the first trimester. There aren't actually huge numbers of regulatory T cells, and the killer T cells are sort of kept out with a chemokine gradient. And many of these mechanisms had been better studied in mouse, but much less in human. And so we are able to kind of shed light on what had basically been more of a black box, basically in the human context. And from basically the background of our group at in structural studies in protein structure, where we are coming from in terms of asking questions about the protein complexes at cell-cell interfaces, was can we use the stoichiometry of proteins that form receptors and proteins that form ligands, and then basically project that data onto cell-cell interactions to check whether all the subunits that contribute to a complex are present, and then predict a specific cell-cell interaction. So that's kind of how we approached it. We integrated protein complexes of known three-dimensional structure as well as protein interactions from other databases, biophysical or biochemical, and then coupled that with a computational pipeline, cellphonedb.org, that we've maintained together with Rosa Venter-Tormer's group and Mirjana Efremova's group over time, and developed further also for projection onto spatial data. So that's kind of the summary of the work on mapping the placenta, where we basically biopsied all the different tissues of this human organ to get a comprehensive cell atlas of a whole human organ with all its tissue microenvironments and cell lineages, to gain a deeper understanding of the maternal tolerance of the fetal cells and the restructuring of the uterine tissue. And so what I want to move on to next is another story from our group in studying an adult organ that's also has multiple different tissue microenvironments. I didn't go into that in detail in the placenta so much, but in the heart, there are probably about 30 different tissues with the chambers, the vessels, the valves, et cetera. And the cell-cell interactions are, both paracrine, and again, we're kind of very interested in immune interactions, but also endocrine, and I'll come on to that. And what that means is that there's both local and kind of long distance signaling, if you like, throughout the body, so through the hormone system. And as well as interaction, of course, with the sympathetic and parasympathetic nervous system that are connecting our brain to our heart and telling it whether to beat slower or faster in a kind of unconscious process that's taking place all the time. So right now you should be super relaxed listening to a fun story and have a very slow heart rate, I hope. And that's determined by interactions with the pacemaker cells in the sinoatrial node that you can see here in gray. And the other tissues that we studied are so the sinoatrial node and the atrioventricular node that contribute to the cardiac conduction system, the free walls of the four chambers, the two atria and the two ventricles that are kind of the muscular tissue of the heart that creates the pumping, and the interventricular septum and apex. And of course, these are connected via the cardiac conduction system, and that system is shown here in this animation. It's not animating, but anyway, it doesn't matter. So the pacemaker cells are here, and they send electrical signal through the Purkinje fibers and the Purkinje cells, and then tell the myocardium in the four chambers how to contract in a harmonious way, basically to pump the blood around the body. That pumping is determined by spontaneous firing of electrical signal through so-called pacemaker cells. And these are amongst the only spontaneously firing cells in the body. They have a kind of internal clock of ion channels that just develops during pregnancy, early in pregnancy, and then just keeps going basically throughout our life until we take our last breath. And it's that that spontaneous clock-like mechanism that keeps our heart sort of coordinated. And of course has inputs from the autonomic nervous system, from the endocrine system, and so on, that sort of speeds up or slows down the rate of the beating. So what we really kind of wanted to hunt for were those very precious and rare pacemaker cells, and they're hard to find kind of in a surgical dissection of the heart, because the sonode is quite a small structure. It's not visible to the plain eye to the surgeon, and it's in a slightly different place, from one person to the next. So those things kind of made it hard, and we hadn't caught that region in our first study of human cardiac cells. But in this second attempt, James Cranley, who is a clinician scientist, PhD student in the group, and now a clinical lecturer in cardiology, and Kazumasa Kanemaru, who is a Japanese clinician scientist, postdoctoral fellow in the group, managed to carefully dissect two hearts from deceased transplant donors that were declined for transplantation during the pandemic. One, they traveled down to London to get one, and then up to Newcastle to get the other, because the transplant was so low during the pandemic that it was very hard to access any tissue. And what they caught were about 50 nuclei that you can see here that had very high expression of a calcium channel and lower expression of a sodium channel that's highly expressed in working cardiomyocytes. And so that gave us kind of the first hope that we might have caught these cells. And then what James did was also curate all G protein coupled receptors and ion channels to sort of expand the cell phone DB database and plot the pattern of receptors and channels of GPCRs and channels here kind of in the columns, and then compare the putative pacemaker cells to other working cardiomyocytes in the atrial and ventricles and neural cells. And what was that cell population had a very kind of signature of channels and receptors with all the typical markers that we would expect, the calcium channel, also HCN1 channel, and so on, to make that clock. And then the other thing that we checked was where those cells are located in a tissue section of a sinoatrial node. And the way we sort of caught the sinoatrial node was by working with a cardiac anatomist and histologist, Professor Yen Ho at Imperial College. And she identified the kind of champion tissue sections for us in tissue blocks that were most likely to contain the node, and then annotated the node by hand. You can see the yellow region here. And then in our Visium spatial transcriptomics data, we used the cell to location sort of Bayesian framework probabilistic framework to identify the pacemaker cell signatures, and found them inside the inner nodal region of this structure, and then with fibroblasts and neural cells kind of in the outer regions. And so that was pretty exciting, because morphologically this region looks homogeneous, but basically with genomics we can see differences within the region and kind of molecular geography of the tissue section to define more fine-grained microanatomical structures. And indeed the pacemaker cells are on the inside, and then you've got sort of electrical insulation and neurals, sort of glial cells hugging the pacemaker cells around the outside. And here we're using HCN1 marker to give us confidence that these are indeed pacemaker cells. And then we identified this new glial cell population that has glutamatergic signaling, sort of maintaining the pacemaker cells, or sort of hugging the pacemaker cells. And in that niche, we identified also special fibroblasts that deposit an extracellular matrix, and then the gene expression signatures of sympathetic and parasympathetic neurons, although we can only see the projections, of course, not the cell bodies, because they're outside the sort of myocardium. And because of this sort of unique molecular kind of signature of pacemaker cells, and because of their importance in drugs that are chronotropic, that are either slowing down or speeding up heart rate, what I asked Kazemaza and James to do is to basically go to our neighbors at the European Bioinformatics Institute, which we were at the Sanger at the time, so that was right next door, and basically investigate whether we can extract all of the approved drugs and tool compounds from the Chembl database, which is a publicly available chemistry database maintained by the European Bioinformatics Institute, and then develop a pipeline for mapping the drugs to individual cells in single-cell RNA sequencing data. And so that basically yielded for the pacemaker cells all of the expected kind of chronotropic drugs, but then it also yielded a very surprising set of drugs for diabetes and obesity. And that was kind of puzzling to us because these included the GLP-1 receptor agonists like Wegovy, Ozempic, and Mounjaro, and so on, which didn't really make sense at first sight. Why would they be acting on pacemaker cells? And the reason is, of course, that the GLP-1 receptor is expressed in human pacemaker cells. This is also a lesson in studying that we need to study ourselves and not mice, because mice don't seem to have this receptor and this signature. But when we looked at the clinical trial data for these drugs, there is indeed a 6 beats per minute increase in heart rate reported on onset of the drugs in the clinical trial cohorts. And so that seemed to suggest to us that the mechanism of action of these drugs on heart rate is through this pacemaker cell, this direct mechanism, rather than through an indirect mechanism by the autonomic nervous system, which had been suggested previously. And of course, there could be, both things could be happening in parallel. But of course, the reason that this mechanism seemed more logical is that the drugs are acting on the hypothalamus and reducing hunger in that way. And so the suspicion was that they'd be acting by the nervous system instead of having a direct effect on pacemaker cells. But what our data suggests is that there is indeed a direct effect, and the referees basically asked us to validate that in vitro, which we did in iPS-derived cardiomyocytes with Michaela Noceda's group at Imperial. And since then, there's also been a porcine study, so pigs basically reported in circulation, which goes into a lot of detail in dissecting basically this mechanism. And pigs seem to be more similar to us than mice because they do have regulation via regulation of heart rate via the endocrine system in this same way. So the summary of this work, or what I want to point out here, is that we define a niche for the cardiac conduction system, and it includes kind of the crosstalk with the autonomic nervous system. So we can sort of expand our cell location from paracrine and sort of long distances via the nervous system, and also even longer distances via the hormone system, which travels all sort of across, coordinating across our organs throughout our body. And also develop the drug to cell framework for that has utility for kind of mechanistic understanding, toxicology, repurposing, and so on. So in this talk, I really want to kind of emphasize the theme of how cell atlas technologies can tell us about cell communication and cell function. So it's not just a descriptive kind of mapping exercise, but there are a lot of predictions and hypotheses about how cells function that we can interpret from the data. So we learn about cell types and cell states, but we also learn about tissue niches and their interactions. And in this story about the heart, what I've also pointed out is that we learn about physiology in terms of, for instance, hormonal signaling, neural signaling, etc. So in the final exemplar of healthy human cell atlas projects from our group, I want to talk about the thymus, because that's my favorite organ. It's the place where T cells are made. And if you remember, our motivation for studying the placenta actually came from T cells and tolerance. And the thymus is the organ where T cells actually develop. Hematopoietic stem cells come from the liver during pregnancy, or from the bone marrow, basically after the end of the second trimester. migrate to the thymus, and then the thymocyte development takes place in a kind of very complex multi-stage process where the cells are trained to recognize or distinguish self from non-self. And once they've completed that training, they colonize the periphery. And of course, T cells are also interesting nowadays because they are sort of therapeutic products in and of themselves in CAR T cells, for instance. And so the question that we were asking with this little organ that sits next to the heart, sort of nestled between the heart and the lung, is the question that we wanted to ask from a human cell atlas kind of tech point of view is, can we actually map the cell atlas of an entire organ? And can we achieve this by modeling multiple modalities together? And so the way we tackled this was by modeling both H&E images, so bright field microscopy, as well as single cell data sets and spatial data sets onto a single nonlinear a single nonlinear axis. And the way the thymus is structured is into lobes. And we can basically use landmarks to mark the edge of each lobe, so the capsule kind of around the outside, and then a couple of key features or landmarks that are conserved and that you can detect across both imaging and molecular modalities, such as the corticomedullary junction, which is the junction between the outside tissue. You can see it here in the inner region of the thymus, and then the medulla itself. And if we use that modeling framework, what it allows us to do is to position tissue section kind of relative to where it sits in this sort of quasi spherical kind of model, if you like. So you can think of this organ axis model where we're using landmarks to position on a sort of nonlinear axis individual data points, whether they're from microscopy, whether they're from single cell, whether they're from spatial transcriptomics or multiplex immunohistochemistry, kind of onto this single axis. And what that allows us to do is, for a given tissue section, which you can sort of think of as a salami slice, we can then position it kind of in terms of the depth through the organ. So if you think of an egg, you've got the egg white and the egg yellow, and if you take a slice, it might be completely in the white, and then it would be either at the top or at the bottom. Whereas if you have a lot of egg yellow, you know that it's sort of going through the middle of the egg yolk. And in that way, we can basically assemble and model together basically the different data sets that we had in this work, which included sort of many different tissue sections of spatial transcriptomics for two sort of developmental time points, second trimester fetal tissue, and then postnatal tissue in infants, which is when the thymus is at its most active. And in that way, basically create a model of the entire three-dimensional organ, at least to a first approximation. And what that allowed us to do was to define kind of in a discretized way, in different layers in the capsule, subcapsular region, different outside and inside levels, the cortex, the corticomedullary junction, and the medulla, where different cell states, such as the stem cell or the early thymic progenitor, are located, and then the intermediate states in thymocyte development, double negative, double positive, alpha beta T entry, and then CD4 helper T cells and CD8 killer T cells, which are then mature. And so we can sort of use this data to define the fraction of cells of these different cell types in the different regions in fetal tissue and in pediatric tissue, and make a model of the journey of the cell from the stem cell to the single positive differentiated cell state, and define in a kind of semi-quantitative way how this journey basically occurs and how much time each cell state spends where in the organ in both the fetus and pediatric tissue. And there were some surprises here in terms of sort of where the early thymic progenitor enters and how quickly and how long it sort of lingers in the medulla before it exits to the subcapsular space here in fetus and in pediatric. And there are differences in the fetal versus the pediatric organ, because during organogenesis, basically the cells are more the cells kind of go to the subcapsular space faster, essentially, and spend more time out here. Whereas in the pediatric tissue, it's the double negative cell that's sort of traveling capsular space. So the learnings from that, and then there were a lot of new insights into detailed cell states and tissue microenvironments in this differentiation, and also in the epithelial cells that are doing that training, telling the cells which antigens are self, and where the stem cells are located, basically. So you might ask, what's the point of all of this? Detailed knowledge of our organs and our differentiation and developmental trajectories. And one of the applications of this kind of detailed cell atlas data is that we can use the information to design and engineer protocols in the dish in a more targeted and informed way to make cells that are basically faithful to their in vivo counterparts. So taking the human cell atlas of T cell development of the thymus, and then using that information about the signals and the transcription factors at the different stages of T cell development to advance thymic organoid protocols in the dish, the things that Chenchu Suo and Dinithi Sumanawera did in using kind of a gene expression trajectory alignment in a Nature Methods paper. So it's one of the examples of applications of the human cell atlas. But I want to just acknowledge the people who worked on this sort of reconstruction of the whole thymus cell atlas. So Nadav Yayon was an incredibly talented postdoc in the group who's now works in the spatial biology discovery research platform here at the Cambridge Stem Cell Institute. Veronica Kedlian was a student and then postdoc in the group, who's now moving to the European Bioinformatics Institute, to the Saez Rodriguez group. Our collaborators, Mus Hanifa, who's been a partner in crime on many immune development projects, the core teams in the cellular genetics and now cellular genomics department at Sanger, John Marioni and Virginie Ullman at EBI, Ron Germain at NIH for the multiplex immunohistochemistry with the IBEX technology, Gigi Notarangelo's lab also at NIH, Tom Tagan's lab in Belgium, and the tissue repositories and pediatric hospitals for the thymi from cardiac resection material from cardiac surgery. And the organ axis, so this was published last year in Nature, and the organ axis framework is on GitHub if anyone's interested, as is all of our software. So I want to finish off with a few kind of stories that talk more about, or that give more insight into the utility of the human cell atlas and practical applications. And one of those applications emerged in the pandemic, where we were faced with a virus that was known to enter the body via the ACE2 and TMPRSS2 receptors. And so what we did across the whole human cell atlas community, kind of starting with the lung bionetwork, which we formed kind of very, very quickly overnight, basically in early 2020, is cells from the different tissues, kind of that you can see schematically here. We asked which cells express those combinations of receptors. And the answer that came back to us was that there are specific cells in the epithelium of the cornea and conjunctiva, so in our eyes, in the specific cells in the nasal epithelium, so goblet and secretory cells, and then in the oral cavity, there are ductal epithelial cells in our salivary glands that have those receptors, and that those tissues are the tissues where most likely there are specific cells that are welcoming to the virus. And then inside the internal organs, there's obviously the AT2 cells, the alveolar epithelial cells that are known already from SARS-CoV-1 deep down in the lung, and then other specific subtypes in other internal organs, including indeed in the placenta and the decidua, kind of for vertical transmission. And so this was giving us insights into horizontal transmission from person to person and also transmission within the body. And that principle is a principle in terms of understanding viral entry that we're using now for other viruses of basically epidemic threats that the UK Medical Research Council and other funding bodies are funding us for. So it gives insights into where could, for instance, zoonotic, or what could be the mechanisms of zoonotic transmission and so on. So that knowledge and information was incredibly important in terms of personal protective equipment and advice around mask wearing and so on early in 2020, where there was still, there were still debates ongoing. It seems ridiculous now, but that's how it was. And the point that I want to make here is obviously that the human cells isn't only useful for pathogen entry factors, but it's also useful as a sort of a lookup table or guidebook for asking questions like where are hormones acting? So where are the hormone receptors? Where are drug targets? We've already mentioned drug to cell. Where are genetic genes associated with risk variants for either rare disease or common complex diseases expressed? Essentially thinking about it as sort of a way of making the human body addressable or transparent, kind of in terms of the molecular makeup of our body. In the last story on kind of applications of cell atlas technologies, I want to move to what I'll call disease cell atlas studies. And of course, disease samples, tissue samples, sort of tumors, infected tissue, inflamed tissue, and so on, have been studied kind of from the even before healthy reference tissue in some sense. But what was, again, during the pandemic, what was incredibly exciting was the possibility of studying tissue from nasal swabs and blood from people who were undergoing controlled infection. And so this was the UK COVID challenge study, which is an experimental medicine challenge study, where healthy volunteers that were seronegative, so there are about 36 people between the ages of 20 to 30 that volunteered to be infected early in the pandemic. And the really interesting thing about these studies is that we can ask what are the early cellular responses before clinical symptoms even come in the nose and the blood. We can also ask, why are there some people who actually defend themselves against infection, so they never have clinical symptoms? And of course, you don't see those people in a normal study because they don't even know that they're ill or that they have any immune challenge that they're responding to. And therefore, can we identify potential mechanisms that confer protection? Shouldn't be infer, should be confer protection. And so in this challenge model, basically the pre-alpha strain of SARS-CoV-2 was inoculated intranasally into a cohort of volunteers, and we were able to raise funding from Illumina and 10x Genomics very, very quickly in the pandemic to study 16 of the total cohort of 36. So the clinical study was described in Nature Medicine by Chris True at Imperial and colleagues, and the single cell genomics study was then published by us later with Chris and Nicole Litchlab and collaborators. And there were three, as I mentioned, so three were there were different sub-cohorts within this. So people who had sustained infection and who were positive across multiple PCR and antigen tests and had cold-like symptoms. People who had a single intermittent positive test and very few symptoms, which we call transient infection. And then people who never tested PCR antigen positive had no symptoms, and we call those abortive infection, even though all of them got exactly the same titer of virus. And the way the study was set up was that we were able to get blood and nasopharyngeal swabs at day -1, so kind of pre-infection, and then day +1, 3, 5, 7, 10, 14, and 28, and study the blood and a nasopharyngeal swab by single-cell RNA sequencing and TCR and BCR and site sequencing. So we had the antibody panels. And so the focus here is really on the immune compartment, but we also were able to study the actual epithelial cells that were infected because we're able to map the viral reads. And so for the three cohorts, the sustained, transient, and abortive, you can see that the pattern of immune cell infiltration is sort of starts at day 5 in the sustained infection. Day 1 already, like a strong response in the transient infection. So even before there are any clinical symptoms, the cells in these people know that they're defending from a virus. And then, the third group is the abortive group, which has very little actually response except for a T cell response, helper and killer, and a depletion of monophils, which could be because they're kind of exiting and or turning into macrophages. We don't really know. If we look for markers that distinguish those people who have abortive infection in the nasopharyngeal swab and blood, there's an innate cofactor that comes up consistently, which is HLA-DQA2. It's very poorly characterized and it's specific to B cells and myeloid cells. So that they seem to sort of B cells and myeloid cells in these people who have abortive infection seem to have a special kind of high or a special state of innate immunity within the cells. And Rick Lindebaum, who is the postdoc who worked on this, is following up on the function of that interesting cofactor that could be somehow restricting the virus. So what this kind of disease cell atlas, particularly with longitudinal data from the same volunteer, the same patient, tells us is a deep understanding of the dynamics of early cellular responses and the differences between the different groups. And also allows us to gain insight for the first time into the sort of pre-exposure immune signature that's associated with protection, which is this HLA-DQA cofactor. And overall, basically, it's this is a paradigm that we're now using in vaccine studies, in other experimental medicine challenges, and we can also use these signatures as a way of sort of timing snapshot samples in terms of where people are in their infection time course. And so just to sort of summarize, that work was, again, massive teamwork between the clinical teams in Imperial down in London, also the HVivo company and the Royal Free Hospital and the UK Vaccine Task Force. And it was a genomics collaboration with Marko Nikolić's lab at UCL and spearhead by Kylie Warlock and Rick Lindebaum, who's now started his own group at the Netherlands Cancer Institute. And of course, massive gratitude again to all the volunteers and the medical staff who were incredibly brave very early in the pandemic when we had little information. So it was a pretty incredible kind of project to be part of. So just to summarize, we are assembling the Human Cell Atlas as a community and releasing reference data objects one by one for all of the bionetworks at data.humancellatlas.org. And this is really an interaction between the bionetworks and the analysis working group, the data science kind of community in the Human Cell Atlas. I mentioned our study of the placenta, and in particular the cell phone DB framework for inference of receptor ligand interactions, the cardiac conduction system mapping and the drug to cell framework, and then finally the thymus and organ axis, and then at the end the sort of UK COVID challenge. I want to mention that the way I hope that we can leverage the Human Cell Atlas in future is to really create a molecular map of the human body in 3D. And of course, we're not going to salami slice ourselves and then get a cell atlas of every single cell. But what we can do is hopefully integrate with other larger volume modalities like hierarchical phase computational tomography or medical imaging modalities and kind of get from a 0-dimensional to 2-dimensional and eventually to sort of volumetric reconstruction of our body. And we have a Canadian Institute for Advanced Research program called the Multi-Scale Human, where we meet twice a year with a small group of fellows and scholars, and there'll be a call for kind of joining this for young PIs, which is a really fun kind of essentially meeting community twice a year to drive this sort of concepts forwards and help each other. These are my disclosures. I work part-time as VP Translational Research at GlaxoSmithKline. That sort of takes up 30% of my time now. And one of the kind of really interesting, exciting things there is that we have a phase 1 clinical trials unit in the hospital here. And we've built up a translational research precision medicine lab that has spatial and single cell biology within the lab, and that can sort of take clinical trial samples in a good laboratory practice, good clinical practice, kind of compliant framework. Like an exciting way that we're sort of bringing the cell atlas technologies into clinical trials. I want to stop there, say thank you, and take any questions.
Thank you, Sarah Teichmann. And I'd love to see if anyone in the audience have any questions, please feel free to just speak out loud.
I could go first. Hi, Sarah. Nice talk as usual. And I'm so sorry my kids are at home with their friends, so you're going to hear some screaming in the background. But one of the questions I had, and I was curious, a lot of the spatial technology that you're using has been focused on Visium. And I'm just curious about the choice. Why not like MERScope or something to identify rare cells? Because we've had some challenges, but I'd like to get your thoughts on it.
Yeah, so no, that was just in those early, or the heart and the thymus projects, but we have used a lot of like essentially Resolve. We haven't used a huge amount of MERScope, but we have used multiplex FISH technologies, Xenium with all different panels and so on and so forth. Immunohistochemistry, multiplex immunohistochemistry like IBEX and Akoya, and also RareSite, which is a one-shot 60-plex commercial platform, so the multiplex protein technologies. For detecting rare cells.
Yeah. I think with a lot of the spatial that we're having issues is that you're getting almost genes that are super specific and single cell become they become double hot, or it's probably the segmentation issues that are associated with single cells. So you're not, like a lot of the good markers that you would identify from single cell RNA-seq are not quite as good in spatial that you're using, especially if you're coming back and designing an informed panel versus an unbiased spatial panel. And so that's where I was struggling with.
Okay. The segmentation is certainly an issue, no question. The segmentation kit in the Xenium has helped a lot for that.
Okay. Yeah, great talk.
But it depends on the cell type, obviously, whether how regular or irregular it is. But I wouldn't say, I would say the markers that are identified in single cell, they generally do validate spatially in our hands, at least in my experience. So I'm happy to sort of take it offline.
Yeah, you could talk. Yeah. They do validate, but there's some like noisiness in spatial. But yeah, I think.
Yeah, it depends on the detailed, whether it's sequencing based or imaging based. I think the imaging based tends to be cleaner, kind of in terms of, yeah.
Should I just go next?
Yes,
Hi,
please.
Sarah. Hi, Sarah. Super
Hi.
inspiring. My name is Ariel Shikanov. I work with Sue
sure.
on reproductive atlas.
And
my
thank
question,
you for your leadership in that, both of you, because we're such an important bio-network.
I appreciate it. Thank you. My question is more about since we sequence deceased donors, and I often get this question, like, is N of 5 or 10 people, is it enough? So I was wondering, how many people is it enough? Like, are we talking about dozens or hundreds, and then how do you really resolve this question? Is like, how do you, like, BMI is a big question, or race is a big question. Age, at least age, we're focusing on reproductive age women. But beyond that, how do you decide, how many people is enough?
So I think it depends on what's the scientific question, and if it's basically just saying like coarse-grained, what are the cell states, or what are the major kind of tissue microenvironments, then the small numbers, generally get all the key, the key discoveries. But of course, I mean, aging is super important in reproductive tissues, right? Just like the thymus, thymic involution is super important. So you want to, I would say in the long term, we do want to have like all the decades of life kind of covered, but obviously maybe that's not the immediate goal. I mean, it makes sense to focus, let's say, on, reproductive age, menstrual cycle, or whatever for uterus, in the short term. But then in the long term, it would be great to have like postmenopausal tissues and so on. You know what I mean? So it really depends on the scientific question, I would say, and also, capturing the diversity of human populations. Again, that's important. And something to maybe think about in the long term. But obviously, tissue acquisition can be really tricky depending on what tissue is you're studying.
Awesome. Thank
Yeah.
you. Thanks.
I actually also have a question. Regarding the like Simon's story, it's so intriguing to see their, I guess, like architectures of the tissue, and also it's like they're kind of like the journey of the cell from stem cell, like time-wise differentiate into like different types of cells, and also spatial-wise kind of like traverse across the space, like to different locations, and potentially staying at a same different time of each location. But from a technology or like data perspective, like spatial data is essentially also a snapshot. Even I guess like even if like we have like multiple time series, it's still they're kind of like distinct cells,
Right.
distinct like spots at different, I guess, time points. Like, is that
Yeah.
from that perspective, is there, I guess, still challenges or like some ways to like reconstruct this like spatial and temporal development?
So you're right, basically, each data point is a snapshot. And so you don't have the kind of in vivo like movie of the cells. And you don't know, essentially, what's the time frame for the development. You can kind of infer that there's more of this cell state in the cortex versus the medulla. So maybe it's staying there for longer, but it's not actually proof, because you don't know whether it's time or abundance of a cell state that's sort of determining that. And so, that was one of the questions actually one of the reviewers asked us. And it was a question about the time for CD4 versus CD8 lineage, basically being in the cortex versus kind of the early decision making prior to entering the medulla at the very early distinction between CD4 and CD8, kind of precursor. And to answer that, we cross compare to kind of imaging data in mouse. So it was 2-photon kind of microscopy in mouse. Yeah, so there are some things that are hard to have from the in vivo human tissue, where you either have to go to the mouse or to a kind of organoid system, yeah, or something like that.
I see.
I don't see any other way. Yeah.
I see. Is there kind of like new technologies that potentially can, incorporate it, I guess, to study this as well as like in general for human cell atlas?
Put an organ on a rig, on a perfusion rig, right? Which is kind of what the transplant surgeons do to keep the organ alive. So that's what you can do, and then do a kind of dynamic sort of analyses. But you need to get the tissue and set up the rig.
Right. I see.
I mean, yeah.
I see. Thank you. Very complicated. Is there not any further question? Maybe we can end it here, Vanessa. What do you think?
Yep, if there's no further questions, we can go ahead and wrap this up because it is at the hour, so I want to make sure we're respectful of everybody's time. But thank you so much for presenting today, and Kong, thank you so much for hosting. I really appreciate it.
Thank you so much. Hi, David.
Thank
Bye.
you.
Thank
Thank you,
you.
everybody.
All right,
Bye.
Thank you, everybody.
Bye.